Hi, my name is Murdo Galvin. Today we'll be talking about how cells synthesize proteins. Proteins are essential biomolecules that play crucial roles in various cellular processes such as metabolism, gene expression, signal transduction, and structural support. The process of protein synthesis, also known as translation, occurs in the ribosomes of cells and evolves into several steps. Step 1 would be transcription. The first step in protein synthesis is the transcription of DNA sequence of a gene into messenger RNA. The process occurs in the nucleus of eukaryotic cells and the cytoplasm of prokaryotic cells. The RNA polymerase, an enzyme that catalyzes the formation of RNA from DNA, binds to the promoter region of the gene and initiates transcription. As the RNA polymerase moves along with the DNA strand, it synthesizes an mRNA, a molecule that is complementary to the DNA sequence. Step 2 would be mRNA processing. Once the mRNA is synthesized, it undergoes several process steps before it is ready for translation. In eukaryotic cells, these processing steps include capping, splicing, and polyadenylation. The 5' and the end of the mRNA is modified by the addition of a 7-methylene guanosine cap, which is the 3' end in the polyadenylated with a string of adenine nucleotides. In splicing, introns, which is a non-coding region, are removed from the mRNA sequence, and the exons, which is a coding region, are joined together to form a mature mRNA molecule. Step 3 would be translations. Once the mRNA molecule is produced, it moves from nucleus to cytoplasm, where it binds to a ribosome. Ribosomes are large macromolecule complexes made up of proteins and ribosomal RNA molecules. The ribosomes have two subunits, the large and the small subunits, that come together to form the functional ribosome complex. The process of the translation occurs in 3 stages: initiation, elongation, and termination. Initiation, the small subunit of the ribosome binds to the 5' of the end of the mRNA, and the ribosome scans the mRNA until it reaches the start codon. The signals of the beginning of the protein coding sequence, the initiator mRNA carrying the amino acid methionine binds to the start of codon, and the start of the subunit of the ribosome joins the complex, forming the initiation complex. Elongation. During elongation, the ribosome moves along the mRNA, reading the codon and bringing in the appropriate amino isolate RNA molecule to add amino acids to the growing polypeptide chain. The ribosome catalyzes the formation of peptide bonds between adjacent amino acids in the chain, and it continues to grow until the ribosome reaches its top cod.